All right, welcome back, everybody. In this lecture, we're going to be exploring our very first branch of psychology that we'll be examining throughout this class. In particular, we're going to be exploring an area of psychology called neuroscience. Now, neuroscience can sort of be considered an area of research on its own or a different branch of psychology depending upon the specific thing that somebody's looking at. But what we're going to be examining in this lecture is something that's really important to both psychologists, neuropsychologists, biologists, and many individuals interested in the basic functioning of the body. In particular, we're going to be exploring a specific concept, or actually, to be more precise, a specific cell that we call a neuron. I'm going to give you a little bit more context before we dive into what exactly a neuron does and how it works, but understand that this specific topic that we're going to be looking at really helps us understand that levels of psychology that we talked about in the previous lecture. Remember back to it, we mentioned how psychologists could study things down to the cellular or even chemical level and all the way up to the societal level. We're going to start small, relatively, I guess, and when we talk about the neuron, and hopefully by discussing it, it's going to prompt this kind of conversation that helps us understand the mind from multiple levels. Before I get too far ahead of myself, let's just start exploring the concepts of the neuron and, well, how we came to understand it throughout the years of research that have been conducted in psychology. So when we look back to our history of psychology, what we see is a science that really came about through the merger of what we call philosophy and biology. These two approaches or ideas of how to understand how the mind and how humans work might not necessarily on the surface seem like two areas that could wed together perfectly. But many individuals for many centuries did have a particular interest in not just understanding basic ideas of how the mind might work, but really trying to find ways to study it in a scientific manner. In the past, when we looked for ways to study the mind in a scientific manner, we were really limited by the resources that we had available to us. If we go back, not only a couple thousand years, but just a couple hundred years, what we see is a world where a lot of the scientific tools we take for granted didn't really exist. So what we had instead when trying to understand the basic principles of the mind were just debates and kind of random approaches that we could come up with to try to see if certain ideas that people could come up with about how the body functioned seemed to test out in real-world examples. And this led to some really interesting ideas about the mind and how it interacted with our body. And if we look back in history, what we can see is that each culture tended to contribute their own ideas and aspects of the mind that persisted for many centuries after these ideas were introduced. One of my favorite examples of this is something that was introduced by the Greeks and Romans and other societies called the humors theory. It was this notion that the chemicals within our body, in particular the fluids that we had a huge impact on things like moods and emotions we were experiencing and energy levels and other kind of overarching aspects of kind of how our mind were shifted on a day-to-day basis. Their argument was that if we had too much of a specific fluid in our body, we might see specific traits, usually undesirable ones, start to emerge. The insistence was that to fix certain things that were popping up, we just needed to identify the specific humor or fluid that was too prevalent and find a way to reduce it, or find the one that was lacking and find a way to increase the prevalence of that specific humor. It led to things like bloodletting, attempts to remove things like black and yellow bile from the bodies of individuals, and other kind of unsavory results. And it gave us this insight into the fact that many people, thousands of years ago, had already realized that there was some interplay between the body and how the mind functioned. But before you get too on board with this idea and start trying to find ways to attempt to remove phlegm from the roommate that you're sitting next to or somebody else that you know, understand that this theory was definitely just that, a theory. There might be some rationale for some of the different components to it, but very few people studying the mind adopt this theory to try to understand individual differences or some of the emotional things that pop up within us. It was, like many of our early theories, something that developed as an attempt to understand the mind and a byproduct of the limited scope that we had to explore it. Another theory that started to become very persistent a couple hundred years ago when trying to understand how the body worked was one that was built on modern mechanics that we were developing hundreds of years ago. It was called the hydraulics theory. It was this notion that our motor system and our sensory system could operate just like a hydraulic engine, where changes in pressure could be the determinants of the sensations that we were getting from the body to the brain, and then the motor movements that we generated from the brain to the body. This obviously required us to understand that the brain was a major component to the mind, and that there probably was a path from the body to the brain and vice versa. But being able to have the tools to look for these units of passage where air or water or some type of pressure could pass was just not possible at the time. So we came up with these ideas, tinkered with how we could prove they sort of worked or didn't work, and just let it be at that. There was also researchers for a long period of time trying to understand this, what we called mind-body problem. Sometimes you'll hear it nowadays be described as the mind-brain-body problem. Trying to tease apart whether or not the thoughts we had, the inner voice that we developed, and all these other things that we consider a part of consciousness could be differentiated from the body. For a long period of time, we just had debates about what this would entail, even though there were some really unique experiments to try to find ways to quantify this idea. One of my favorites was a researcher who would actually have individuals lay on very specific scales when they were on their deathbed, and he would try to monitor their weight down to fractions of ounces to see if their weight would change as they passed away, arguing that if they did lose a little bit of weight, it was actually their soul leaving the body. Now, these research studies were riddled with problems and, well, limited by a lot of the things that we just didn't have hundreds or thousands of years ago. But they really highlighted how many individuals, even before we could be scientific about studying the mind, were really enamored by trying to understand this thing that we call the mind and what allows it to function. And as science started to advance, people started to find ways to explore this idea in a much more scientific manner. Now, the specific moment in time where researchers were finally able to start better understanding how the mind was capable of doing what it did on a biological level came about in the mid-1800s when biologists and other researchers were finally developing the first tools to give us really robust, interesting images of different systems within our body. As we started to classify things like our skeletal system and our respiratory system and other critical elements that allowed our body to thrive, researchers started turning their attention to something that we were already starting to call our nervous system. And their arguments were that as we perfected these tools, these microscopes and other types of dying systems, we'd eventually start to be able to find the cells that allowed the brain to communicate with the body and vice versa, and that really made up the critical matter of this really important structure to our nervous system that we call the brain. For a long period of time, as we were developing these tools, people were sort of forced to just postulate what these cells would look like and how they would communicate with each other. And eventually, a name, a term, started to be used to describe these cells. And they were called neurons. The person first being credited with this name was the theorist named Heinrich Wilhelm Waldier. Waldier, like many other individuals, surmised that these neurons would be very plentiful in our brain and would be the things, like other cells that we had in our circulatory system and other parts of our body, that would allow this nervous system to function. Once we started talking about these cells, people started postulating how they would exactly work. One of the champions who really explored this concept of the neuron before we were able to actually image it was a gentleman named Ramon E. Cajal, who published something called the Neuron Doctrine in his attempts to better explain how these cells, if they did exist, could come together to allow the brain to do all the elaborate things that it did. Cajal proposed that there would be millions or even billions of these cells within our body, and they would be connected to each other, either physically or really close to each other so they could communicate in some means. And that this communication process would have to happen in fractions of a second for the mind to be able to do what it does. Now, Cajal's idea was not just a random thing he came up with. There were lots of advances in biology and research that suggested that these neurons had to exist. We just hadn't been able to image them yet. But the doctrine that he came up with to explain the function of neurons was really poetic and beautiful at the time, and it really spurred on interest by biologists and other scientists, even chemists, to try to be the one to show images of these neurons and give proof to these doctrines that had started to be pushed for a number of decades. And the person who was able to first find these neurons was a gentleman named Camillo Golgi, who developed his own staining technique that involved the use of a specific silver alloy that we now call the Golgi staining technique. And in 1873, he started publishing his first images of neurons that look much like the ones you see in the background on this slide. As Golgi perfected his techniques and others have improved upon it, the images that we started to develop of these neurons became more plentiful and, well, more precise, and it gave us a better understanding of not only how these neurons might potentially function, but how they were able to allow our mind to do all the things that it did. And eventually, over time, as we got better with our techniques, psychologists were able to verify a lot of the ideas that were presented by Cajal and other theorists at the time. We now know that within almost all of our bodies, we have roughly between 80 and 100 billion neurons populating the majority of our nervous system. Having more doesn't necessarily mean that your brain or mind is working better. It just tends to mean that you've got more clusters of neurons in specific areas. What we tend to see when it comes to where these specific neurons are located is most of them are populated in something that we call our central nervous system. That's usually the brain, spinal cord, and some of the cortical structures that are connected to those things that we really think of as the main hub of our nervous system. But please understand that in addition to roughly 80 billion neurons being located there, we have another 10 to 20 billion neurons extending to our body, so we can not only make sense of the world around us, but find ways to react to that world as well. And these neurons, oddly enough, do a lot of the things that Cajal had surmised. They connect to each other in these microscopic spaces or gaps that we call synapses. We have all of these different structures moving in different ways that allow very specific types of functioning for each and every neuron. And they do come in different shapes and sizes to do some of the specific things that we need to have done in different parts of our body. And if we want to understand all the different things that these specific cells make up, we have to understand the specific function of each cell first. And that's what we're going to dedicate the majority of the rest of this class to. Just covering the structures of these cells that we call neurons, then looking at how they communicate with each other and bringing that back full picture so we can then spend some time looking at the brain and the central nervous system in our next class. So let's start a journey here where we explore these cells called neurons that were discovered roughly 150 years ago at this point. What do neurons look like? Well, neurons look a lot like a large number of our cells in our body. In the center of each neuron, we have this thing called a cell body. And within that cell body, we have a hub called the nucleus. This nucleus contains a lot of the critical information that allows these cells to not only survive, but do the things that they're designed to do. For neurons, this specific cell body is critical for these cells to not only pick up on information from the environment, but to commit that communication process that is essential for all of our neurons to really do the things that they're designed to do. And connecting to this cell body are these arm-like structures that are really critical for the receiving process in communication that happens between neurons called dendrites. Some cells within our body have just one dendrite reaching out into a specific area of our body. Other neurons can have a multitude of dendrites reaching out to sometimes different parts of the body, other times multiple cells that might be communicating with this specific neuron. But regardless of how many dendrites each neuron has, their design is always the same. They're there to pick up on some specific message that, if they get enough of it, will activate this cell to communicate with adjacent cells in the body. Now, there's a lot that goes into what triggers a dendrite. You learn about things like resting potential and action potentials and all this other stuff if you move on and take future neuroscience or MCB classes. But in this class, since we're just in Psych 1, all we have to understand is that dendrites are picking up on information from either adjacent cells or certain structures in our environment that are being sent to the cell body so the cell body can decide if it's going to fire or not fire, or commit what we call an action potential or not. And if the cell body does decide to, well, just for simplification purposes, say fire, well, this starts a chain of reactions that occur along another critical component to each neuron in our body that we call the axon. In particular, if a cell decides to fire, the first change that happens within this cell occurs at something called the axon hillock, the part you see highlighted there in the upper left-hand corner of this image. This axon hillock is a critical area where the axon, which each cell only has one of, meets the cell body, which again, each cell only has one of. And a specific physical and chemical change starts to take place. In particular, when this action potential occurs, this axon hillock allows specific ions, and we're going to talk about this a little bit more in the next slide, to start coming in and leaving the cell. And this message is propagated along another critical structure within the neurons called the axon. And it passes all the way from the cell body of this cell down to these branches at the end that we call the terminal buttons, or sometimes you'll see them listed as you see here as the synaptic terminals. They're essentially the ends of the cells where sort of the out message goes. Each neuron, again, only has one cell body, only has one axon. But once the message reaches the end of the axon, it will often branch out along multiple terminal branches to different synapses that could be for just one cell or multiple cells. And it's in this place where the communication, the next step of communication, will take place between neurons. To understand this better, let's go to the next slide. So how do these cells communicate with each other? How is a message sent down an axon all the way up to the terminal buttons, and then what gets a cell to signal another one? As I mentioned earlier, we really didn't have an idea of what allowed these communications to occur between neurons until just about 150 years ago. We had this idea that there would probably be a small space between these cells, and they would find a way, either through physical or chemical or electrical interaction, to trigger an adjacent cell and get it to react in some way. And we now call this microscopic gap between these cells either a synapse, a synaptic cleft, or sometimes you'll even hear it listed as a synaptic gap. And the way these cells communicate with each other is through a very specific chemical that we call neurotransmitters. Now, it's probably important to note at this point that these chemicals are actually abundant in our body in a variety of different forms. If we're talking about them being used in cellular communication in our nervous system, we call them neurotransmitters. Other times, if these chemicals are floating through our bloodstream and activating specific body parts, we call them hormones. It depends on what spot and what types of chemicals we're looking at that kind of determines the name that we're looking at. But when we are talking about neuron-to-neuron communication, we call these chemicals neurotransmitters. And what essentially happens when one cell communicates with another is in these terminal buttons, or sometimes what we call synaptic vesicles, like you see here in this image, these chemicals, these neurotransmitters, are stored and then released when this thing called the action potential occurs, or firing occurs. And those neurotransmitters, those chemicals, sit and linger for a fraction of a second in this synaptic cleft or synapse or whatever we're going to call it for this specific moment. And they linger in a way that allows some of them to activate or sort of inhibit the adjacent cell that's connected in that synapse. If a cell releases a neurotransmitter that does enough to get the other cell that it's connected to then commit an action potential or to fire, we call that neurotransmitter an excitatory neurotransmitter. If the specific cell sending a message to the other one is releasing a chemical that actually prohibits the adjacent cell from being active, and there are ones that do this, we call that specific neurotransmitter an inhibitory neurotransmitter. Now, excitatory neurotransmitters and the excitatory action is usually a relatively intuitive thing for most people. We understand the idea of needing lots of excitatory messages, right? We want one cell to communicate with another so we can activate a whole bunch of different things going on in the mind. But it's important to highlight when we talk about these communications that inhibitory message has a lot of value as well. When we look at the mind and all the cells that make it up, what we need to identify is that out of those 80 to 100 billion of them that we have going on, most of them are not and should not be active at any given moment in time, especially when we look at the brain. For our minds to operate in an effective way, what we need is not only billions of neurons working together, but only working when they're supposed to. I know you might have seen some silly movie along the way that suggests that human beings are only using a small percentage of their brain at any given point in time, and if we turn it on, suddenly we'll have superpowers or have the ability to understand things in different ways. I guarantee that a lot of that is just garbage nonsense. For our brain to function effectively, we need to turn a lot off. And that's a key aspect to these inhibitory neurotransmitters. They turn off large portions of our brain, large portions of our body when they're supposed to be turned off. This allows us to do things like sleep, focus on specific elements of the environment, essentially prevent our mind from racing in ways that it's not supposed to race. This is also a critical player in our motor systems as well. If we were sitting in an in-person class, I'd have us do this really weird activity where I ask you to raise your arm while having your fist pushed against your collarbone or your chest. And then I ask everybody when they're doing this to touch the top of the shoulder of the arm that's bent in front of them. And what I ask everybody to do is kind of identify that regardless of how much you exercise, you should feel muscles being flexed in that upper shoulder, indicating that there were lots of excitatory messages going from the brain to other parts of the brain and eventually down the spinal cord and out to the muscle fibers in our shoulders, creating all these excitatory messages telling those muscles to not only flex but stay flexed for an extended period of time. But as those muscles are being flexed, it's also critically important to note that we've got other signals coming from the brain all the way down to our arms that are screaming at the muscles on the bottom of that arm to stay relaxed, to not activate under any circumstances. Yes, there's a lot of excitatory messages that are traveling from the brain down the spinal cord and out to places, but eventually, when they reach the muscle fibers, they are screaming an inhibitory message to those muscles, telling them to turn off, to not be activated. This is a critical component to not only us being able to do that random thing of putting our arm in our chest and raising our elbow so we can kind of feel one muscle being activated while the other's being turned off, but they're critical in any aspect of balance and force that we're able to commit. Again, though, this is just a minor blip in all the inhibitory messages that are being sent throughout our body at any given point in time. And these messages, in conjunction with the excitatory ones, allow our brain to do really all the amazing things that it does. If you understand that much, that's huge. But if we understand how neuroscientists know so much about the brain, we have to dive a little bit deeper into this action potential process that we mentioned earlier. As I mentioned a few slides ago, when a cell decides to fire, what it does is it starts to commit both this chemical and mechanical change that starts from the cell body in this very specific spot called the axon hillock. And that message propagates itself along the structure of these cells called the axon until it reaches the terminal branches and eventually gets out to those axon terminals or terminal buttons where the neurotransmitters are released. We know what's going on in the synapses, what happens at those terminal buttons, but how does a message get sent from the axon hillock all the way to the terminal branches? Well, it's first important to note that when a cell decides to fire, this is an all or none thing. Neurons do not whisper or yell. Right? Neurons are just sending a message down the axon once they've determined that they need to fire. If we want to talk about the strength of a signal, when we hear something loud or something soft, that's dictated not by the strength of the signal being sent by a specific neuron. It's determined by the number of neurons that are being activated and sometimes even the pattern in which they're activated. Each cell, when it decides to commit this action potential, does it in the same way every single time. And again, it starts at the axon hillock, where there are tiny, microscopic pustules that are actually allowing ions to come in and out of the cell. When a cell decides to, we'll say, fire, what happens is a tiny pustule at the axon hillock opens up just enough to allow a whole bunch of ions to come flowing into the cell. Now again, this is Psych 1. This is not our biological psychology class. This is not an MCB class, so we do not have to memorize the specific ions coming in and out. We do not have to memorize the resting potential and the changes in electrical charge as a byproduct of the ions exchanging. You will get to that, I promise, if you get into neuroscience classes. For this class, just understand that a pustule opens up. You have a whole bunch of ions that come rushing into this neuron as a byproduct of changes in charge and density of ions. And a reaction to those large number of ions coming rushing in is another door to kind of balance things out right next to the first door. Opens up and allows another collection of ions to go rushing out of the cell. Now, I always, to kind of give students an image of what this might look like, ask people to imagine a club or a bar that's very popular with their bouncer being there, not allowing many people to come in, and tell them, an action potential is like that bouncer suddenly saying, everybody go in, I don't care. And everybody comes rushing in, overcrowding the bar. That triggers another bouncer in a different part of the bar or club to recognize the overflow, so they open up a different door, and it causes a bunch of other individuals to quickly rush out, sort of balancing the pressure and maybe balancing even the vibe, we'll say, of that club or bar. It's exactly what happens at the axon hillock. But it's also important to note that when that second pustule or that second small microscopic gap opens, what happens is another bunch of ions start rushing in a different door that opens up. And these channels continue to kind of open and close in this reactionary pairing process all the way from the axon hillock to the terminal branches, eventually down the terminal branches to those synaptic clefts, where there, finally, instead of having these ions come in and out, you have the release of these chemicals that we again call neurotransmitters. These exchanges of ions, the chemical processes involved, even the physical processes that involve the kind of action potential, are really cool. And I encourage you, if you haven't studied these things, to maybe learn a little bit about them from the text, and maybe consider taking some of our extra bio classes that we offer, because it is really exciting stuff. But just in our class, understanding these basic principles and the byproducts of it, the changes in electrical charge that occur inside and outside these axons, is sufficient to understand what psychologists use to understand how the mind works at the cellular level. And also understanding this can help us understand some of the tools that neuroscientists and psychologists use that we're going to be discussing in our next class. But hopefully from this, you now know how the action potential occurs. We'll revisit this a little bit more when we talk about another thing. But now that we've covered that, we can explore some other really critical things that we want to know if we want to truly understand all the things that neuroscientists have looked at and the things that help us better understand the biological principles of the mind. Another thing that's really critical to understand in a class like this is that within those synapses, there's more to it than just activation or excitation/inhibition occurring. Yes, the activation thing is the main player, right? That's the thing that we talk about in neuroscience to understand how cells communicate with each other. By, again, either saying fire, fire, or don't fire, don't fire. But after that communication's taken place, for these cells to function correctly, for things to be able to work at the speed that they work, we also need other functions occurring within these small microscopic gaps that we again either call synapses or synaptic clefts. Another thing that happens so these cells can communicate very quickly and effectively is a process that we call reuptake. when reuptake happens, these terminal buttons or synaptic vesicles that we talked about earlier that originally contained the neurotransmitters can actually open up again, and if there is lingering neurotransmitters in the synapses, they can suck that neurotransmitter back in. And this provides us with a lot of utility. One thing that's very helpful and really allows these things to function well is that this reuptake process prevents these neurons from having to produce large numbers of neurotransmitters every time they want to communicate with an adjacent cell. Another thing that allows these cells to do is communicate with adjacent cells in relatively rapid succession. Instead of there being confusion about whether or not a neurotransmitter is lingering in a synapse because of something that happened half a second ago or less than that, what these neurons can do is communicate, and sometimes in spans of milliseconds, with adjacent cells. And this can be critical in not only maintaining specific neuron activation, but in determining certain components of the message that's being conveyed from one cell to another. A classic example of this is in hearing and vision, something we'll talk about in future classes. In addition to reuptake, helping clear out those synapses, another thing occurs oftentimes when a neurotransmitter has been released. It's called diffusion or metabolization. This is when specific enzymes and other chemicals actually can pass through these very small microscopic gaps that we talked about earlier and actually wash away or kind of disintegrate, or deteriorate, I guess would be a better term, the neurotransmitters that were emitted. This, in conjunction with reuptake, again, really helps the cells communicate effectively with each other. There's exceptions to this. I mean, there's always going to be misfirings, and then actually, when we get into developmental psych, we'll talk about how these misfirings happen more often and happen in more places as we get older. But for most of us, this process works pretty seamlessly for the majority of our neurons in our body, and it allows them to communicate in very effective ways that go beyond just the excitatory/inhibitory message that we kind of think of when talking about cellular communication. There's other cells that also help in this process that are worth mentioning. I think your book finally gives them a little bit of credit as we've been progressing through different editions of the text. I always bemoan the fact that for a long time it wasn't mentioned in the text. I think they're covered now, and they're called glia cells, or sometimes you'll see them listed as glial cells, depending upon the book that you look at. These glial cells, come in a multitude of different forms and do a multitude of different things, but their primary goal, regardless of what they're designed to do specifically, is to help neurons within our nervous system to communicate with each other better. Some glia cells, like astrocytes and even occasionally oligodendrocytes, are specifically designed to remove the waste, the dead cells or the different components of our body from a multitude of different systems that are inhibiting communication between neurons in the way that we'd like to have them work. We oftentimes have debris and dead cells and other things that will prevent our neurons from working the way they're supposed to. And a lot of these glia cells are specifically designed to help kind of streamline that communication process when these things occur. Another thing that some of these cells have the ability to do is actually increase or enhance the communication process that occurs from one cell to another. So far, we've mentioned this action potential as being a cell-to-cell thing. But if you think about it in the grand scheme of things, you got to remember, we have roughly 100 billion cells in our body. And many times, when a message is sent from one cell to another, it's sent in conjunction with a bunch of other cells that are communicating with a bunch of different cells. This is again what allows us to determine things like magnitude or strength of the messages that we have being sent all over the place. And oftentimes, for these cells to communicate effectively, we want to make sure that the messages being sent are happening in a synchronized way. One very cool type of glial cell that exists in our bodies are these cells, which are pictured in the lower right-hand corner, called oligodendrocytes. They actually have the ability to wrap the fatty substance contained within them around multiple axons, not just of one cell, but of multiple cells. And by doing so, what they can do is get the message, the action potential that's going down that axon of not just one cell, but multiple cells, to be synchronized so that when a message is released into a synapse to either just one cell or multiple adjacent cells, the release of that neurotransmitter can be really kind of overlapping within a millisecond or even less than that, depending upon how effective these things can be. And that synchronization of these cells is really effective when we look for kind of cellular communication and not only efficient, but kind of reliable communication from cell to cell. Now, not all oligodendrocytes do this. Some just wrap around these axons for a different purpose that we're going to talk about in a second, but that synchronization is something that I do always like to just spend a second talking about because it has a lot of utility. If we were in class, we'd even do a little activity to highlight how effective it is to be synchronized when doing things like clapping versus just everybody clapping on their own. But you just understand for now that it's great, and the way it works is, well, something you can explore in future classes, but the end product is something I want you to be familiar with. The other thing that has a really interesting kind of end product of understanding how it works is somewhat challenging, is this process that we call myelination. It's a byproduct of either oligodendrocytes wrapping around these axons or another specific type of cell that we call Schwann cells, also a glial cell, wrapping their entire bodies, or their entire cell bodies that is, around a subsection of the axon. When these Schwann cells or oligodendrocytes do this, they don't cover the entire axon of a neuron, but what they do is they create this sheath, this kind of fatty section along this axon that serves for two different purposes. One of the purposes it serves is to help protect those axons, which are not necessarily brittle, but really critical components for neuron communication. There's not much research that suggests that if we do have some damage to an axon, that'll somehow magically grow back or be able to retain its function if there is damage to that axon. Having these fatty substances wrapped around neurons can help with that. But the thing that we typically think of when we talk about myelination is the fact that that fatty substance allows these cells to communicate a little bit more effectively by having multiple wrappings around these axons and these little tiny spots that we call nodes of Ranvier. These nodes, or tiny gaps, are very special critical parts of these axons when they commit the action potential that we talked about earlier. If a cell is not myelinated, as we mentioned earlier, that opening and closing of specific channels has to occur throughout the entire axon. And it takes a little bit of time for that to take place. I mean, we're talking about fractions of seconds for it to happen, but fractions of seconds added on to fractions of seconds do eventually start to build up and create situations where, well, communication from, say, the toe to the brain and back down could add up to maybe even a half second to a full second, depending upon how developed we are. If we have this myelination process occurring, though, that communication that we talked about earlier, the opening and closing of channels, doesn't actually occur along each spot in this axon. Instead, it just happens at the specific nodes. Now, how those chemicals pass through those what we call myelin sheaths, and what allows these nodes to be activated the way they are, is again something you'll learn about in future classes. I promise you'll get there if you take those. But just understand for this class that by having these skip spots, essentially, these cells communicate much, much faster. And this allows for our minds to do some of the amazing things we start to be able to do as we age and our cells become more and more myelinated throughout our body. Have you ever wondered why you can do things faster than, say, a 5 or a 6-year-old? Part of it's because you got a lot more neurons working more effectively with each other, but another part of it's because of this myelination process. A message coming from one neuron to another in your body tends to travel faster as a byproduct of this myelination. It's a really cool little thing that we can use to understand how the brain works, and also how certain issues can pop up with the brain that we'll talk about in future classes. Now that we've covered those things, there's one last thing I want to discuss before we leave. And that's something that we've mentioned previously in a very simplified way. If you remember back to the communication process between cells that I mentioned, I discussed how most cells either send an excitatory or an inhibitory message. This would sort of suggest that the neurotransmitters that are producing that excitatory or inhibitory message probably come in just two forms. Right? You got your excitatory neurotransmitter and you got your inhibitory neurotransmitter. You don't need more. Well, it's not actually the case. What we've seen through research is that neurotransmitters actually come in a wide variety of different forms. And oddly enough, neurotransmitters tend to be linked to specific neurons that are found in specific parts of our brain and different parts of our nervous system. And yes, some neurons do produce multiple types of neurotransmitters, but more often than not, most of the neurons in our body are predominantly producing one type of neurotransmitter, and they're responsible for one specific type of function within that part of the body that is using that chemical in a specific way. And once neuroscientists started to discover these things and recognize that it's more than just inhibitory and excitatory messages at the chemical level, well, this ushered in a new era of exploration that we're still on right now, trying to better understand these chemicals that, again, we call neurotransmitters when we're discussing how they work in those synapses between our cells. And just so you have a taste, a sense of some of the things that we've looked at, I thought I'd list two of the more prominent neurotransmitters in our body, and ones that are really important to just have a basic understanding of as you're starting off and just recognizing kind of the application of this stuff in our everyday life. One of the ones that you might have heard of before in weird ways is a neurotransmitter called dopamine. If you have heard anything about dopamine, it's probably that it's linked to rewards or positive emotional experiences. You might hear a neuroscientist or a biologist talk about how when somebody compliments you or when something good happens, you have dopamine just pumping through your system. That is just the worst way to describe this specific neurotransmitter, but I guess it sort of gets at what it's typically linked to. So again, when we talk about neurotransmitters being released, it's not that these things are just floating through our bloodstream or that they're kind of randomly being spewed all over the place. Instead, when something triggers a huge increase in dopamine levels, it means that all the neurons within specific regions of the brain utilizing dopamine to activate other cells are, well, more active, and they're releasing more dopamine into those synapses. Now, in our next class, we're going to talk about specific brain structures like the amygdala, like the hippocampus, structures that definitely utilize dopamine in occasional moments. And when they do it, they typically do it in a way to trigger parts of the brain and the mind that are linked to positive emotional experiences and also motivation. So when a researcher says you've got this onslaught of dopamine happening in your brain because something good happened, just know that when that good thing happened, it triggered lots of activity in different parts of your brain so you could better understand what led to that thing, better be driven to do that thing again, and I'll just better appreciate that thing while it's happening. And this might lead you to think, well, I want all of my dopamine cells to be active all the time, but understand that could actually come at a cost. If you've got other areas of your brain that are pumping out lots of dopamine all the time, you can start unintentionally activating other areas of the brain that aren't supposed to be active. A lot of our sensations and our kind of early precursors to perception are also dictated by dopamine levels of activity, cellular activity of cells using dopamine in other parts of our brain. If you've got lots of dopamine using cells in those areas firing when they shouldn't, you can start to experience hallucinations or even symptoms that are very indicative of a specific type of mental health disorder that we're going to talk about near the very end of the class called schizophrenia. This is where specific drugs used to lower levels of dopamine have been designed. They can either help with kind of controlling schizophrenic symptoms, hallucinations, or even addictive issues that sometimes come with too much dopamine in some of our reward or positive experience areas that we talked about earlier. But again, we want to make sure that we're careful with restricting too much dopamine activity, because if we do, we can start to break the critical connections between certain parts of our brain. Individuals that struggle with Parkinson's tend to have less and less dopamine in some of our motor areas of the brain that are critical for not only the activation of specific muscles eventually, but the coordination of specific pathways to different parts of our body that we, again, tend to take for granted until they're not working effectively. When we combine this with all the stuff we talked about earlier, hopefully you can understand the complexity of our different neurotransmitters and the fact that just one neurotransmitter can be linked to a lot of things. Another one that's worth mentioning, and we'll talk about this at a couple different junctures in the class, is a neurotransmitter called serotonin. Now, this one, much like dopamine, is typically tied to one specific thing, in particular mood and psychological wellness. Numerous specific disorders have been tied to the prevalence of serotonin in specific regions of our brain. Again, prevalence of serotonin means the amount of activity of specific regions of our brain that use serotonin as their means of communication. What we see is that really low levels of serotonin tend to be linked to things like depression and anxiety and other problems. And we also know that not only can these things impact our mind, but certain things we do can impact the production of these things and create this sort of cyclical pattern. We know that hunger, sleep, arousal, energy levels, all of these things are very closely tied to the serotonin levels in our body. And there's certain things that we can do in terms of how much sleep we get, how much exercise we get, how much sun we see, that can be tied to changes in our serotonin levels in different parts of our body. And understanding the link between this specific neurotransmitter and some mental functions, again, gives us some insight into how important understanding neurotransmitters on a general level is for people in the field. Now that we've wrapped that topic up, or at least given me a taste of that topic of neurotransmitters, I think we're really well situated so we can understand other aspects of neuroscience and this kind of branch of biological psychology in our next class. So we're going to wrap up for today, but know that we're not quite done with the bio stuff yet. We focused on neurons, focused on how they work and all the different components to it that you would need to know at an intro level. Now we're going to move on to what these things come together to make, in particular the parts of our central and peripheral nervous system that we'll talk about next. So for today, thank you very much for listening. Appreciate the attention. I wish you all the best, and I hope to see you soon. Take care. Darn it, forgot I had an extra slide. Well, I just gave you all the information that you needed in the last slide. Again, take care. See you soon.